Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolon, I'd like to welcome you to Metabolomics as a leading technology in biomarker discovery. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I'd like to introduce our presenter for today. He is Dr. Brian R. Kepler, PhD, Associate Director of Project Management, Global Pharma and Biotech with Metabolon. Welcome, Dr. Kepler. The presenter ball is yours.
Thank you, Elizabeth. Today, I'm going to talk to you about metabolomics and how Metabolon has really pioneered the development of metabolomics and become a true leader in metabolomics offerings, not just for pharma and biotech, but for academic and government institutions as well. I'm also going to put a biomarker twist on it. Although metabolomics and Metabolon's technology is applicable to a number of different goals and objectives, mechanism of action, disease elucidation, patient stratification, things like that, it's going to be more of a twist on biomarker discovery towards the end. The first thing I want to talk about is Metabolon. We are a company headquartered in Research Triangle Park, North Carolina, founded in 2000. We have a CLIA certified CAP accredited labs. We also have GCP capabilities with our targeted analysis. Although we are headquartered in North Carolina, we do have a global presence. We recently acquired Metabolomic Discoveries in Potsdam, Germany, late last year, now having an established EU operations near Berlin. We also have technology platforms established in China and Qatar through licensing deals. And presently, globally, we have about 220 employees. These are not solely analytical chemists. As I'll go through the infrastructure of Metabolon and how we deliver our results, you'll understand that it's the full analysis, bio-IT, statistics, and interpretation that goes along with our output. So our goal is really to deliver quality data that we are confident in so that we can give you confidence in finding actionable insights through our interpretation. So first look at what is metabolomics and how does it fit into the central dogma of the sciences. So DNA, RNA, protein, all the other omics. I'm sure most of you listening are involved in doing genomics, transcriptomics, proteomics. So metabolomics is one step closer to the phenotype. Whereas something like genomics or DNA gives you what the risk of a disease might be, gene suggesting, is there a mutation? It's actually the penetrance of that is most telling of how phenotypically relevant it might be. And metabolites are those things that are the actionable information. They're most proximal to the phenotype. Now, in addition, looking at metabolomics can bring you a connection to not only what's happening endogenously, but also exogenously with your diet, your environment, even the microbiome. The medications you take, all these things can influence the microbiome and the metabolism. And we do have insight into looking at all that and making the connections and how these influence the phenotype. Now, I mentioned this before, the technology and the infrastructure. So I'm going to talk to you about many different offerings that we have at Metabolon. Again, all things metabolomics, looking at small molecules, small metabolites, and biochemicals. We have a biochemical database that is truly unmatched in the industry in terms of its coverage and comprehensive look at different pathways. We look at a chemocentric approach, so we're distilling these ion features confidently down into precise chemical entities. And we do this with tier 1 identification, and I'll talk about what that means later. And then using our patented software. So a lot of what goes on here is taking that data, a large amount of data that's generated, and really making sense of it. What metabolites correlate with that data, reducing the data, eliminating the noise, and giving you confident data. And then finally, the bioinformatics and expert interpretation that's involved. So really taking it a step further, providing you not just raw data, but actionable insights and interpretation by our biochemists here, such as myself, that we look at your data and basically act as a FTE on your behalf to explain what the data means, put it into biological context for specifically what your matrix is, what your design is, what therapeutic area you're looking into. Now, before I get into the technology, I want to make it clear that This is going to be more looking at biomarkers towards the end, and I just want to make it clear that the technology is applicable across the continuum of different types of research, basic research, in vitro, looking at preclinical models, looking at clinical patients as well. And again, as I mentioned before, the goals and objectives are vast in terms of, do you want to look for a biomarker or safety profile, mechanism of action, target engagement, different things. So I ask you, if you'd like to find out more information towards the end, you can ask questions, but also contact us if there's some other information that we can provide. So now looking at the technology. So the first thing we're going to look at is the global approach, an untargeted look, trying to cover all of the different classes of metabolism. We'll move into some more targeted approaches and applications that we have after this. So the first thing is simple biochemical extraction. So whether you're looking at a circulatory biofluid, such as plasma or serum, or looking at urine or feces or any type of tissue, whether it's from human or animal or cell pellets with in vitro, it's a very similar extraction where we're basically precipitating out the DNA and the RNA and the protein so that we just have our small molecules in our sample that we are injecting across the platform. The platform itself has 4 different methods. They are all ultra-high performance liquid chromatography in tandem with mass spec. The mass specs are Q-exactive accurate mass. And the reason for these 4 methods, you can see some of them are optimized for positive ionization, some for negative ionization. We also have a Hillock column to capture the polar molecules. And the reason for this is because different metabolites are going to have different biochemical properties. They're going to elute faster or slower. They're going to be more prone to be positively ionized or negatively ionized. They're going to be greasy like lipids. And by running these 4 methods, we're able to capture all of these different classes of metabolites. So it's truly a global approach. Now, when we acquire this data, again, there's a tremendous amount of noise and data reduction that's needed to take place. And Metabolon has developed proprietary and patented software to do this on a high throughput but very precise and accurate fashion. This is also highly regulated by our QC, by which our data spectra analysts and data curators are looking at the calls made by the automated software, making sure that we are confident in the calls, that the integration is done properly, that the quantitation can be done accurately between groups, and to give you, again, a confident, rich data output. The next steps that we do in-house are the statistical analysis. This is a number of different techniques which would be dependent on your specific study design and your questions. This is all given in an output of a client data table, where you have all your raw data, the heat map, typical heat maps, red being statistically up, green being statistically down, plots as well. And then we also have a Metabolink portal, which allows you to look at your own data and play with the stringency of the p-value, or look at the different effects of group A versus group B, for example, on a pathway visualization, or doing pathway enrichment. So we allow you to have access to this portal, and myself or someone on my team also uses this portal and these types of tools to do the interpretation. So again, using Metabolon's institutional knowledge, we have over 1,000 publications, over 7,000 completed studies with hundreds of clients. Chances are that something that you're looking into is something we've done before. We can give you some information on how well it turned out and give you suggestions for optimizing a study to achieve one of your own objectives. This is the Tier 1 metabolite identifications. I mentioned this once already. This is very important regardless of who you do metabolomics with. The Metabolomics Scientific Initiative has developed this. This is an initiative, a committee run by KOLs in pharma, in biotech, academics, government institutions as well. And they come together to generate this tier system about identification confidence, basically. So when you acquire features, ion features, when you get tentative structures, putative identification, or a validated identification with an actual reference standard, this allows you to jump up in the tiers, Tier 1 being the best. Now, the important thing to note that is that a lot of companies, a lot of academic institutions, they vary around Tier 2, Tier 3. There are some that can do Tier 1. The Broad Institute, for example, has some Tier 1 identifications, but their coverage is not nearly what we have. A quick note on the coverage, we're looking at a library, comparing all of what you see in the library to about 5,000 named metabolites. Now that doesn't necessarily mean you'll see 5,000 metabolites in plasma or serum or your tissue. As an example, a representative example of plasma, you might see about 1,000 metabolites on this platform. So again, excellent coverage, well represented in all the different major pathways, which I'll talk about. So Metabolon, about 95% of what we see, we are at Tier 1. So Tier 1 identification, we have an ideal standard, a reference standard which we've compared it against, and we can be completely confident so that you can be confident in all these calls. About 95% of what we show you is that Tier 1. There are some metabolites simply for which we don't have a reference standard or we have not been able to synthesize yet. Those are going to be Tier 2, but we actually label those metabolites that are Tier 2 so that you know a simple a reference standard is missing. But Tier 1 on almost everything, and again, regardless of whether you're doing metabolomics on your own or searching it out, it is very important to be confident in your data. So this is a very important thing that really sets Metabolon apart. A little bit about the technology, the history. I mentioned before, Metabolon in the field of metabolomics, we've truly been a pioneer in helping to move this technology along. If you look back in 2005, median relative standard deviations in plasma for all metabolites detected was about 48%, right? Not too good. But as you can see, we've gone through many generations of the platform over the years, as depicted by the dots here. These are basically milestones of hardware upgrades, software upgrades, a number of different things in technology and bioinformatics that have allowed us to now have the platform that we have. Last year, our median relative standard deviations were single digits, less than 4.5%. As you can imagine, the confidence that you can have in the data with numbers like that. And the coverage, as the RSDs go down, the coverage is going up. You can see this is saying about 2,500 metabolites in a plasma sample. That's including the global and a complex lipid panel together. So just looking at global, I said it's about 1,000. And then about 2,500 if you do a complex lipid panel on top of that. So again, very good coverage. Now, in terms of, in order to give you an idea of what types of metabolites we're going to be seeing, again, it's looking at all the major pathways. This is a map. You can't see the individual metabolites, but it's a map of all the major pathways in mammalian biochemistry. The plant biochemistry is going to be a bit different. We actually have a separate map for that. But all mammals, you know, horses, rats, mice, dogs, humans, it's all going to be very conserved looking at what you see here. There's also some microbial metabolism that's not represented here, but we also have insight into that, and I'll talk about that later. So this map shows you all the major pathways, and each blue dot is actually a metabolite that we routinely see in a plasma sample. So as you can see, there's thousands of them, about 1,000, and you can see good coverage in all the major pathways. So what are those major pathways? These are all the major pathways. So looking at amino acid metabolism, carbohydrate metabolism, energy, lipids, cofactors and vitamins, nucleotides, even some novel metabolites, drug metabolites that patients might be taking. And then finally at the bottom, the established microbiome. As you can see in each one of these major pathways, there's a number of different intermediates and subpathways that come along with those. And with a library of 5,000 metabolites, you can see there's going to be hundreds of metabolites in each one of these subpathways. So very good coverage. And just to emphasize the bottom here, the one that's boxed, the established microbiome. So the library itself, that 5,000, it's mainly mammalian metabolites, but it is also plant. Like I said, we do a lot of ag bio work, but also microbiome, so microbial-derived metabolites. And the importance of that is the fact that in our human health, our microbiome is very important and very highly linked to disease. So by having these metabolites that are solely produced by the microbiome, so they are converted by dietary nutrients, for example, converted by the microbiome, and then they're recirculated through the body. You can detect these in not just feces, but blood and urine as well. And having insight into these established microbial metabolites can be very informative. So that is a look at the global approach, what we just talked about on the left-hand side, the blue, the global metabolomics, casting the wide net, really a hypothesis-generating approach. The reasons you might use this is that you haven't narrowed down where a drug might be having an effect or what precise pathways might be most affected in a treatment or in disease versus healthy, whatever your objective or study design might be. We also provide a number of targeted approaches. So instead of the untargeted global approach, we have a lipidomics approach. This is actually the complex lipid panel. So although the Metabolomics has a number of lipids, which I showed you on the previous slide, things like phospholipid breakdown products, some bile acids, free fatty acids, endocannabinoids, steroids, a number of things in the global approach. Because of the nature of lipids and the sizes and the requirement for different extraction protocols and different methods for identification, we've broken out a complex lipid profile into a targeted panel. And that's what I said assisted that number to be 2,500 for total metabolites that can be identified in plasma. If you add this complex lipid panel, which I'll give you more detail in the next slide, if you add that to the global. Now, if you want to look at more targeted metabolites than just global or lipids, we also have a number of targeted metabolite panels that I'll show you on the next slide. Ones that are already developed and ready to go, or ones that we can custom develop for you as well. So the targeted panels here, again, I mentioned the complex lipids. I'll show you some detail. It's actually on the next slide. We can simply look at fatty acids, short-chain fatty acids, which are very relevant in microbial metabolism, bile acids relevant in microbial or host metabolism. We have a cholesterol metabolism panel, stratum corneum lipids, and sebum lipids. So these two lipid panels were actually broken out because the lipids that you find in the skin and the sebum being the oily substance that's secreted from the skin, they're very different from all lipids in other parts of the body. So we have specific panels for these. We also have a panel called AccuGFR, which is an algorithm looking at specific metabolites to more accurately generate a glomerular filtration rate that is more precise and accurate than eGFR. We have a C4, which is reflective of bile acid metabolism. And then we have a metal ion panel looking at the metal ions, which can be run in a number of different matrices, not just blood or urine, but a number of different tissues as well. So these are ready-to-go panels that you can look at. Again, not as broad as the global, obviously. We're not looking at 5,000. Each one of these panels has its own specific number of metabolites, and if you contact us, we can go into detail on that. Now, if you have, let's say, a customer, and this is often the case, has already done metabolomics with us and wants to focus on a set of 5 metabolites, or simply one metabolite, and they want to do fully targeted quantitative methods on that. We can start that from scratch. We can do method validation, development. We can do it under the qualifications of research use only or under GCP. And it can basically be any metabolite that we can find a method for in terms of finding stability and finding a reference standard. So the abilities, it's really endless capabilities that we have here, and we would just ask you to contact us, and we can have a call about with our targeted assay team to discuss those options. So the last piece of technology I'm going to show you here before we get into the biomarker part of the talk is this complex lipids. So again, the global has its own set of lipids. The complex lipids are going to be these more higher ordered structures. Things like triacylglycerols, diacylglycerols, the monoacylglycerols. It also has some redundancy with the global. There's some free fatty acids and some single myelins that might have some overlap. But we also have PCs, PEs, PIs as well. You can see all of these different classes actually constitute, if you look at the number of species, because this is a fully looking at composition and speciation, you can see the number of species add up to over about 1,100 different species. You can see the triacylglycerols, again, with 3 fatty acid backbones, you're going to have 3 fatty acids on the backbone. You have about 519 unique triacylglycerol species alone in this panel. So it can be very informative, again, for applications of what you might use in terms of the global metabolomics or complex lipid panel or targeted in your first approach. Really, my best advice would be just to contact us and we can have a call on that. So moving towards how our technology can be and has been applied, again, we're going to focus on the biomarker aspect, that type of goal and objective. The discovery metabolomics is really where a lot of people start. It's the best way to start in terms of looking for a biochemical biomarker. It's really the untargeted approach that I've been talking about. If you look at the global metabolomics and the complex lipid panel together, you'll see about 2,000 metabolites. And it'll really give you a good idea and a good start to narrow down in what pathways in your objective and your study design, whatever your comparisons might be, what pathways are being affected if you don't already have insight to that already. After the discovery, again, the validation approach moves to the validation moves to the verification approach, and we have, again, the capabilities for the targeted assays. So a couple quick case studies that I'm going to show you here. Each one is just on one slide. There's quite a long story that goes with each one of these, and there's publications that are indicated here that you can go to that we can provide. But I'm going to show you the true observation that stood out. There was a lot that allowed the clients to move forward and really identify a good biomarker for their needs. This first one is looking at systemic lupus erythematosus, or SLE, looking at for biomarkers in serum in either healthy or SLE patients. So SLE, it's a very diverse autoimmune disease. Reliable and specific biomarkers have really not been found, and they hinder clinical management and the development of new therapies. So the goal, of course, was to identify something that stood out in these SLE patients versus those that are healthy. So serum was analyzed in this study, and if you look down at a high-level look at a small snippet of the data that we provided, you can see things are ordered. They're by metabolite on the y-axis here. You can see each of the metabolites. Each patient is on the y-axis here, and they're grouped by healthy and SLE. So you can see, looking at these metabolites, a lot of these are very low in healthy individuals, as represented by the green, statistically significantly down in green or up in red, whereas they're up in the SLE patients. So that tells us, obviously, one thing at a high level, that these patients are very different, and we need to look at the meaning of these different metabolites. So taking that, again, a step further, providing the actionable insights that our interpretation can yield. Another thing to note before we move off of the data here is there's a couple SLE patients that are all green here. Now, these are patients that have been diagnosed with SLE, but you can see they look more like the healthy. So you can see just by a simple high-level look, and if you were to take a look at this data and look in the paper, you would see that these individuals have a much less aggressive form of SLE, which is why at this level they're looking more like the healthies. But if we take a look at what most of these patients are looking like, elevations in a lot of these metabolites, some of them being things that are reflective of inflammation. So if we look at the key findings here, this is a look at serum leukotriene B4 levels. Healthy, very low levels. SLE, significantly increased. 5-HEAT, another metabolite, a product of toxin LOX conversion of fatty acids. And you can see, again, the healthy, very low. SLE is very highly elevated. Both of these metabolites, along with some of these other ones that are here in number 3, are reflective of inflammation. So you can see, and it's not, this would be expected in a disease like SLE, an autoimmune disease, that you're seeing reflections of inflammation and an inflammatory response. And it's really this leukotriene B4 that really stood out. If you see the scale here, it is much higher in the SLE patients in this global approach versus the healthy. And a verification of these results, if we look at the validation here in an independent assay, independent cohorts, and also using a disease mimic, looking at rheumatoid arthritis and RA and comparing that, SLE in this verification is not only, again, a good biomarker, LTB4 is not only a good indication of a biomarker for SLE versus healthy, but it even separates out, it distinguishes between 2 different autoimmune diseases, rheumatoid arthritis and SLE itself. So what Metabolomics has offered here in this study was the identification of new biomarker candidates that can be taken forward and further validated by the client, and also develop insights that could help to form the basis of new therapeutic strategies. So again, this is a very quick look at the main highlight from one of these case studies and the papers here in the +1 publication from 2012. Just to note, if you go to our website, there's a number of other publications you can search for with different keywords. And again, contacting us, we can talk about these other examples as well. So I have one more case study before the end here. Again, another biomarker case study. This actually has a microbiota twist and a renal toxicity twist to it. So a microbial-derived metabolite was actually identified as a marker of renal toxicity, and also off-target safety implications as well. So the background of this one, a compound was found by a client to cause acute renal failure. So they have a compound, it has its phenotypic response, a desired response, but it also causes kidney failure. They know that the toxicity was not related to the mechanism of action. This was based on some knockout work done by the client. So they were looking for biomarkers of renal toxicity. The study design is shown here in Figure 1. They had urine and plasma collected from mice, from CD1 mice, male mice, and they dosed them either with low, medium, or high dose. They collected plasma over a time course, or they collected 24-hour urine together. Now again, there's different ways and different tools to look at the data at a high level. You can look at the heat maps, you can look at PCA plots, random forest, hierarchical clustering. We can provide all these types of things. This observation here, this example is looking at z-score plots. And basically each one of these plots is looking at the different doses and time points for the plasma. And if you take a look at vehicle at 1 hour versus 24 hours, you see some changes. Vehicle 1 hour versus the high doses, you see some changes, but you're seeing most of the changes, most of the shift in these standard deviations in the high dose at the longest time point. But the beauty of this study design is that we have where you're really affecting the system, and you'll see that from the data, but you also see earlier time points. And when we look at the specific data in a more granular view, such as really the main metabolite of interest here, 3-indoxyl sulfate, you'll see that it's really the earlier time points that are precipitating this buildup of this toxic metabolite. So going from a high-level view at the z-score, really picking out, and again, just focusing on not all of the data from the study, but focusing on what the main takeaway was. It was with this metabolite called 3-indoxyl sulfate. And it's developed from dietary tryptophan. It comes in, it's digested, it goes into the gut, it's converted to indole. Indole itself can go back into the blood to the liver to be converted to other things. The gut bacteria can convert it however, to IPA, convert that to the blood. And it's really the conversion of this indoxyl, which is sulfated in the liver, to indoxyl sulfate, 3-indoxyl sulfate. Again, it's converted by the microbiome, which can't be done by the enzymes in the human, so it is truly a microbial-derived metabolite. When that gets recirculated into the blood, that's when the problem arises. This microbial metabolite, it's elevated at a high dose, much more and more dramatically than the standard renal markers as well. So here's a look at the data. 3-indoxyl sulfate in the red here, you see it much higher elevated than both creatine and urea. Again, bona fide markers in the literature of renal response. So we're picking those up, which is good proof of concept, but you can see 3-indoxyl sulfate is much more elevated. So what was the client able to do with this data that we provided to them? Well, they developed a targeted assay to screen this compound and other samples along with backup compounds. And what they found was, again, with their toxicant, with their drug that causes this undesired renal failure, you can see it in red. Over time, you see it increase, just like we did in the global approach here. But they have a backup compound in blue. You can't see it because it's so low, actually, but on the right hand of each one of these time points, you see a blue bar. That is actually a backup compound that has almost the same pharmacophore as their lead compound, but one moiety is different. It's a little bit different, and you can see it in the paper if you go to this publication here. But the simple change of one little moiety allows it to still be efficacious and potent in the way they wanted. Maybe not as good as their lead, but it lacked this toxic effect in the kidneys for these mice, and allowed them to basically kill a compound that would never have gone forward, and also identify a compound that they could then take further. Now the last slide here is basically just want to drive some points home. Again, we are the infrastructure of Metabolon is well developed for high throughput metabolomics, using our rigorous processing, monitoring, our QC steps to provide high quality data, our informatics and our interpretation to provide actionable and biochemical insights. Again, as I mentioned before, there's a number of different applications. If, during this talk or in the future, if you have an example of where you think metabolics might fit into your workflow in order to move your needle forward, in order to satisfy an objective or a goal, we would be happy to speak with you. My contact information can be seen here, or you can go to metabolon.com to get in contact with anybody, and we will be returning your emails shortly. Thank you. And I'd be happy to take any questions that anybody might have.
Thank you very much, Brian. Our first question for you, what sample types can be used with this technology? For example, dried blood spots, microbial samples, and so forth.
Yeah, great. That's a good question. I mentioned a number of different tissues that could be used, but this gets to a good point in terms of the dry blood spots. So flash frozen, stored at -80 samples, shipped on dry ice for plasma, serum, tissues. That was the norm up until recently. That, of course, is all still good and viable and produces high-quality data. But in addition to that, we have actually done a lot of R&D in terms of determining whether dry blood spots that are not stored at optimal temperatures can still be used to glean actionable data. And the answer is yes. We've done a lot of research and validation on this. Dry blood spots, which doesn't require as much volume, can be easily acquired in regions where deep freezers are not available. They can be easily acquired from small infants, much easier than getting actual plasma or serum. And because of these reasons, because of the market need, is the reason we developed this. So this is about ready to go. So if you have dry blood spots, I urge you to, again, have a conversation with us, and we can give you more detail on that. Another type of matrix that was previously unable to be used because of the limitations of metabolomics are formalin-fixed paraffin-embedded tissues. So this is also a recent update in Metabolon's abilities, whereas these samples can now be used. Again, I won't go into detail with it now, but so in addition to naive fresh tissue, if you have flash frozen paraffin-embedded FFPE tissues, I encourage you to talk about the abilities and the utilization of those samples as well.
Yes, I think you actually answered the next question, but just for clarification, you did say that formalin-fixed paraffin-embedded tissue can be used, correct?
It can be used, that's correct. It is applicable to the technology.
Okay. And we have a question from Dave. What kind of sample volumes do you require, and how fast is the throughput on those samples?
Yes, the volume requirements or weight requirements will depend on firstly will depend on the offering that you take, whether it's the global or the complex lipids or a different panel. In general, these are very low. I mean, they'll range between, let's say, for a biofluid, whether it's urine or plasma or serum or even media from culture, generally between, and again, it's going to depend, but between, I'd say 30 to 200 is the range. So some of these volumes are hard to come by with things like small animals, where we can use smaller amounts. Optimally, if you have patients where you can provide 200 microliters for full global and complex lipid analysis, that would be optimal. But again, a discussion about what you have and what types of things or limitations we might see with smaller volumes, those are typical conversations that we have with a lot of clients that we're able to discuss those in more detail about what we can offer. So basically, so very small volumes. The weights, it's basically the same rule, about 20 milligrams to maybe 100 or 200 milligrams, again, depending on the types of offerings that you need. And then in terms of the turnaround time, again, it's going to be very specific to the type of offering, the level of interpretation that you might have, whether it's just a targeted assay. Again, we can get things out very quickly. We can have things expedited. Again, that's a question that I would urge you to contact us, and we'll have a business development executive and someone like myself get on a quick teleconference, and we can discuss those and give you more accurate ideas of those numbers and those timelines.
All right, thank you. John asks, do you conduct SCFA ratios, and if so, which SCFAs do you report?
So yeah, short-chain fatty acids. So the short-chain fatty acids are going to be represented on that specific targeted panel. I believe there are 8 short-chain fatty acids on it. I don't have it in front of me, but it's going to be all the, I don't have it in front of me, and I'm sorry, I don't want to get it wrong. That is available on our website. Again, if anyone wants to get those specific lists, we can simply email you the PDF. But there's about 8 short-chain fatty acids, and that is fully quantitative. So from that quantitative data, you would be able to get short-chain fatty acid ratios.
All right. Our next question, how many compound standards do you use in your lipidomics panel?
Yeah, so the lipidomics panel, which is fully quantitative, and you can see about 1,100 different lipids. Again, I don't have the precise number of exact standards that we have. It's not going to be 1,100. There are certain ways and techniques we use to use, say, one standard for a certain class of different lipid metabolites. That is definitely something that I would bring in our lipid analytical team on to have a call with anybody who wants more information on that. But we can provide that information.
Excellent. Another attendee asks, with the reference standards that you use, are you able to provide the absolute concentration of a particular metabolite?
Yes, on the targeted panels. So the global approach, which I told you, looks at 5,000 named metabolites, compares to a library. That is not running 5,000 metabolites every time. So we're not running a standard every time. So that's a relative quantitative approach. That global untargeted approach is going to be relative quantitation, very highly validated, precise, accurate in terms of fold change values that we return. The targeted approaches, which includes the complex lipid panel, the other panels as well, the bile acids, short-chain fatty acids, that list that I had, those are all fully quantitative. So yes, you would get the precise, whether it's a micromolar amount or whatever it might be, if it's a liquid or a tissue, you would get the precise amounts, yes.
And I believe there was a follow-up question to that about how many standards are available currently. Didn't you mention 1,100?
We have standards for all 5,000 named metabolites in the global. The complex lipid panel, which can identify 1,100 different species, has, I don't know how many standards it is. I believe it's not 1,100 because we can use some metabolites to cover, some standards to cover multiple metabolites. I don't have the exact number of standards that we use for that complex lipid panel, which you can see about 1,100 species. But again, I can, that's easy to answer if somebody wants to contact us offline, and we can get on a call and talk about that in detail.
Perfect. What are some of the top applications of your technology for pharma companies?
Yeah, sure. So we work, and I work personally with all of the major global pharma companies, really. Biomarkers is one application, mechanism of action. We work, and again, we work with the full gamut, the full continuum of research in some of these pharma companies, preclinical, clinical, also R&D, looking at in vitro as well. An important thing to note about culture, cell culture, we can look at the cell pellets as well as the spent media that they're in, which gives you excellent insight into what's going on. And cell culture can be a great model, depending on what stage of R&D you're in, can be a great model for many things, specifically mechanism of action. We have some great case studies on that. I'd be happy to share with anybody who wanted further information on that. MOA in vitro. A number of other things as well. I think one of the earlier slides mentioned some of the things, not just biomarkers, MOA, but patient stratification, target engagement. Really, there's a vast array of different applications in pharma.
Good to know. I'd like to very much thank our presenter for today, Dr. Brian Kepler. I'd like to thank our sponsor for today, Metabolon. Most of all, I'd like to thank those of you who came and spent this time with us. We very much appreciate you choosing to be with us. We know you're very busy, and we say thank you again for your attendance. On behalf of Cambridge Health Tech Institute and Metabolon, thank you so much, and have a great day.